Hello, Biology 360 fans, or should I say people who happen to be enrolled in Biology 360 this semester in Malone section. This is going to be your lecture capture extraordinaire for Chapter 10 and part of Chapter 11. So let's begin. This is just a little background of the history of DNA and its discovery, well, not really discovery, but identification as DNA, as the genetic storage unit for all heredity. So I'm not going to ask you guys any dates, but just please take a look at it and get a realization of how recent most of this information has come about. I know it seemed like the 50s was a long time ago, but really it wasn't that long ago. Okay, don't forget this. This is a nucleotide. This is all 107 stuff. Okay? Please, please, please be able to recognize a nucleotide by phosphate sugar base. Right? Phosphate group. Our sugar, depending on whether it's DNA or RNA, is either ribose for RNA or deoxyribose for DNA. And our bases are how we name DNA, right? Either A, G, C, or T. And remember, if it's RNA, there's uracil, U, and no T's. So that is review, and you should be good with all that stuff. Okay. So here's two new things that we didn't talk about in my 107. I'm not sure if your prerequisite class discussed these. If they did, great. It's just a review. If they didn't, then this is all new for you. Please make sure you can explain these experiments and understand who did them and why they did them and what they found. So it'd be really great if you went through the book and read these, especially if you don't understand after I'm done with this quick tutorial on these two experiments. One is generally known as the Griffith Experiment, and the other is either Avery or Avery, McLeod, and McCarty. Okay, for the Griffith Experiment, it's known as the transformation of bacteria experiment. It essentially told us that the substance, there is a substance within bacteria that can genetically transform another bacteria. Okay, so there's something that can move from bacteria to bacteria that changes the genetics of a bacteria. Okay, and so that was the first indication that there's some element within a living organism that determines its hereditary fate or what it expresses. Now we know it's easy. It's DNA. It's the genes. It's how you express them, right? Genotype leads to phenotype. But at this point, no one really knew the answers. And so let's walk through this experiment. There are several different types of bacteria here. One is considered virulent. A virulent bacteria is something that can kill you, right? And so if you infect a mouse with a virulent type of bacteria, guess what happens? The mouse, feed up, dead mouse, okay? After you do an autopsy, yes, you can get these bacteria recovered. That makes sense. It's a bacteria known to kill things. You give it to a mouse, it kills it. In part B, a non-virulent strain of this bacteria, non-virulent means it doesn't kill you. Guess what? You give it to a mouse, the mouse survives. And when you do the autopsy, there's no bacteria in that mouse. That suggests that the mouse's immune system cleared all that bacteria, right? That's what our immune system's supposed to do for us. Okay. In part C, we now take this virulent, right, the type 3S, so again, this is the virulent, this is supposed to kill us, right? This bacteria is supposed to kill. And if you actually cook the bacteria, kill all of those bacteria so they're dead, and you inject it into a mouse, well, guess what? The mouse survives, because all the bacteria was dead before you had it in there. So when we do the autopsy, there's no bacteria recovered here. So all three of those make sense. All three of those are considered controls. What are controls? Controls are something you compare your experimental to. So part D is actually the real experiment that happens in this event. And so part D, they take live non-virulent. So the live guys, the ones that are alive, don't kill you, right? They're the good ones. And they mix them with the junk from the bad ones that are heat killed. So we take some of this stuff and we mix it in here. These are all dead, right? But all their chunks are in there. So all the membranes, all the DNA, all the RNA, all the proteins, all of that is in this mix. And we mix it up here with the live non-virulent, right? The ones that are not supposed to kill you. And then we inject that into our mouse. And guess what? Our mouse croaks. Okay, great. Right? We compare that to this one where we just added the junk and it lived. So it can't be just the junk that killed it, because we showed right here that the mouse lived with just junk. And it can't be just the non-virulent here, because again, B was our control for that. We add the non-virulent, the thing croaks. Or the thing survives. If you mix those two together, our mouse dies, and after autopsy, what we get back is this virulent type of bacteria. Now, we did all the controls to make sure everything we moved from up here actually was dead. So it's not just that we were crappy scientists and we didn't kill them all. Okay? They did this many times, many replicates, and every single time they came out the same, and this virulent bacteria was recovered. Okay, this suggests that something in this goo is what causes the virulence. Okay, so something in the goo from the bad bacteria, the virulent one, got into the non-virulent and caused it to become just like it. Now we know it's the DNA. The DNA from the bad bacteria got into the good bacteria, the non-virulent one, and changed its phenotype, right? You add genes to an organism, change the genotype, that changes the phenotype, okay? And so that was the first evidence that we can genetically transform a bacteria from one phenotype to another. At this point, they did not know what that transforming factor was. They just knew it was something in this goo of the dead stuff, of the dead virulent bacteria. That is known as the Griffith Experiment. Okay, so later, in the next set of experiments we're going to talk about, Avery, McLeod, and McCarty wanted to know, what was it? What was the stuff in the goo? Okay? What is the transforming principle? And so they did a series of experiments to eliminate one thing at a time and then determine whether their bacteria was still virulent or not. Okay, so first they start essentially where the other experiment stopped. They take that virulent bacteria and they heat it up and they boil it, right, and they essentially kill it. Homogenize means to stir it up and then filter it, okay? And then they take this goo, right, so this is essentially the goo, and they want to know what in that goo actually causes that phenotypic change in those bacteria. And so they make a couple guesses, because of course you have to make an educated guess, and they say, well, what if we get rid of all the RNA, right? What if we just, no more RNA, we're going to enzymatically destroy all of the RNA, okay? And then we're going to add the goo that's left to, again, our non-virulent stuff and see if it can become virulent. And when they do, guess what? The virulent stuff is still there. Okay? So whatever's in the goo was still able to turn non-virulent into virulent. So it can't be RNA. RNA cannot be the transforming principle because there wasn't any in there. So it has to be something else in that goo that causes that genotypic and phenotypic change. In number 2, they decide to destroy all the proteins, right? Do the same thing, infect their bacteria, and what happens? Yes, again, non-virulent becomes virulent. If there's no proteins around and it still works, then it can't be the proteins doing that transforming principle. In their last set of experiments that we show here, they decided, what should we do? Let's destroy all the DNA. So if we get rid of all the DNA, right? Destroy it with specific enzymes that only destroy DNA, does not destroy RNA or proteins or lipids or any of the other stuff, and then take the rest of that goo and infect this bacteria. Guess what? It does not transform. The non-virulent stays non-virulent. Non stays non, right? There was no transformation, nothing changed. That suggests that what they destroyed in the original goo is the transforming principle. Okay? And so they came up with the end that it is DNA. That's what's in the goo that causes that phenotypic change when it enters that other bacteria. Okay. The last experiment we're going to talk about is the Hershey-Chase experiment. And again, I want you guys to know these by name and by what they did and what they showed, right? We did virulent and non-virulent bacteria in the Griffith transforming experiment. What did we find out? Yes, there's something transferable. We don't know what. In the Avery experiment, again, we used the virulent and non-virulent bacteria to figure out what the transforming principle was. What did we find out? It's DNA. And then in the last one, just to be sure, since they did experiments where they got rid of lipids and they got rid of all kinds of stuff, but we supported the fact that it's DNA by getting rid of it, but we didn't directly show that we could put DNA in something and cause it to be transformed. We showed that if we don't put it in, it doesn't get transformed. That's not as strong of an experiment or direct as doing what Hershey and Chase did. Okay, so first let's start off with a little background on these guys. I want to make sure you guys understand what a radioactive label is. Radioactive labels are things that give off radiation. And why is that a good thing? Well, it's generally not a good thing for us, but it is a good thing for experiments, especially way back when we couldn't follow molecules with fluorescence or something that's not dangerous. So if you radioactively label something with a radioactive isotope like P32 or S35 or iodine or one of the other radioactive elements, you can follow the molecules with which they're incorporated, because as radioactivity breaks down, it gives off light. And you can trace light at that time by spots on film. Right? We don't even have film anymore, but anytime light hits film, it makes a dark spot. So only where radioactivity happens, it makes a dark spot. You can also use a Geiger counter, which actually makes noise. I don't know if you've watched any movies from the 80s where they have Geiger counters, and it makes clicking noises whenever there's radioactivity. Okay, so it's just a way to follow different molecules. So in this experiment, we use a radio label of P32 for DNA and S35 for protein. Why do we use P32 for DNA? Why do we use S35 for protein? If you don't know what DNA is made up of or protein's made up of, please go back and review 107. You're killing me! Okay. Right? Phosphate in DNA, phosphate sugar base. Right? P32 gets incorporated. S35 or sulfur, right, is in cysteine. So most proteins have some cysteines in it, so you can radio label those. So let's walk through this experiment. And what we start with is two different halves, okay? And so we're going to split this experiment essentially right down the middle. And on the left side, we have soaked everything in S35. And on the right side, we've grown up bacteriophage in P32. And so if you grow up these bacteriophages, or these viruses that infect bacteria, any of their proteins will incorporate the S35, right? Here, if they're being grown in this radioactive sulfur, and if they're being grown in radioactive P32, the phosphates and the phosphate sugar base of the nucleotides will be radiolabeled with phosphorus, okay? And so then we can follow those two radioactive molecules by their giving off light or their noise on the Geiger counter or using radioactively sensitive machines. Okay, so on this side there's only S35, there's no P32, and so the phage should all take up S35 in their proteins. Okay, at that point we infect E. coli, or a bacteria, right? The bacteriophage is a virus that infects E. coli, gets inside. Okay, and on this side we do the same thing, but we're using P32, so now all the DNA on this side should be labeled. All the protein on this side should be labeled. Right? Again, we infect bacteria with this guy. It gets inside. And then we actually use a real live blender. Right here is an actual blender. Now remember, this was a long time ago. They didn't have a lot of super fancy machinery, so they actually used a wearing blender. So in some cases, this is called the wearing blender experiment. If it was a Black & Decker blender, they would have called it the Black & Decker or a KitchenAid, but it was the brand Waring. So the wearing blender is sometimes what this is known as. And so they actually blended up these bacteria, which would remove all the virus that didn't get inside, right? Everything would get off the, if it didn't get inside the bacteria, it would be sheared away or blown away from the surface of the bacteria, and only the viruses that got inside would stick around. And so then they take a centrifuge right here, and they spin by centrifugal force all the heavier stuff down to the bottom of a tube. Okay, and so if we draw out what would look like a tube, right, heavy stuff is going to pellet down here. So big stuff like a bacteria. Anything floating around in the liquid stuff would be up top. Okay, and so you could essentially separate, right, here's the bacteria and here's the any phage that didn't get inside. Okay? And so then all we have to do is look at where did the radioactivity go. If we're looking at the protein side, right, this side, and if protein were the agent that these bacteriophage used to transform a bacteria or get inside and make new virus, the genetic material of the virus, we would find all the radioactivity in the bacteria, right? That makes sense? Because if that's what's the genetic material for a virus, it would have to get inside the bacteria in order to replicate itself. And guess what? There was no radioactivity in the bottom here. No radioactivity in the bacteria. All the radioactivity was up here in the liquid, suggesting that protein is not the genetic material of a bacteriophage. On the other side, we're back looking at this, and now if the genetic material was DNA, the labeled radioactive DNA, it should be in the bacteria at the bottom. The heavy stuff, that should be radioactive. And when they looked at the bottom of the tube, guess what? That was radioactive with P32. And nothing in the liquid part was radioactive at all, suggesting that the DNA had to get inside the bacteria to do the genetic transformation and allow that bacteriophage to reproduce itself. And so this was a direct experimental evidence that DNA, at least in bacteriophage, is the genetic material. Okay, and so now we know that the phage genome is DNA, and it's up here in the head of this phage, and it looks like a little lunar landing molecule, and it sits right on the bacteria. And what we see is that phage gets in, dumps its DNA inside, and then, remember, viruses are not living things, so they have to use the cell that they're inside of, in this case a bacteria, to make more of themselves. So they use the machinery for transcription and translation, and they build new viruses and burst out new viral particles. Okay, we're back. It's time for the Watson and Crick model of DNA as a double helix. Okay, so what we need to know about this that we maybe haven't talked about in the past is that the ratio of A's and T's are always 1-to-1. There's always an equal number of A as T and an equal number of G as C, right? Yes. Okay, and so if we look at the base composition of DNA from many different sources, we're going to see that this is always true. And why is this always true? Because of complementary base pairing, right? The A to T ratio is always close to 1. The G to C ratio is always close to 1, right? That makes total sense because A's always pair with T's and G's always pair with C's. Now, what a lot of students get confused on, that doesn't mean that the number of A's and T's is equal to the number of G's and C's. All of those don't have to be in a 1-to-1-to-1 ratio. So if we look at the first one here, E. coli, it pretty much is, right? 1-to-1 to 1-to-1. There's about equal A's and T's as G's and C's. But as we can see in yeast, they're very AT rich and very low in GC content. That's totally fair game. That doesn't matter as long as the A's and the T's are equal and the G's and the C's are equal, because that's how the complementary base pairing works. But an individual organism can be very AT rich and GC low, or GC rich and AT low. That doesn't matter. Okay. We also want to talk a little bit about X-ray diffraction. This comes back to Watson and Crick determining the structure of DNA. And again, our book goes through this whole process. You don't need to know this, except that this diffraction pattern, based on understanding this, was what allowed Watson and Crick to realize this was in a repeating pattern, that the pattern repeated itself. They used the information about G's always equaling C's and A's always equaling T's to come up with the structure of the DNA molecule. And so before we get to that, we're going to do a little question, and you're going to answer this, screenshot and upload to number 21. Right? If you extracted DNA from a bacteria and found that there was 37% cytosine, how much percent guanine does this species automatically have? Okay, so please answer that question. You can circle it, take a screenshot and upload. And then the next one, the DNA was extracted from cells of this same bacteria. It has 37% cytosine. What percent thymine does this species have to have? Again, please answer this question. Take a screenshot and upload to number 22. All right. All the rest of this stuff is review. Please make sure you know this. If you don't know this, it's not going to be good. Right. So I'm going to go through this pretty quickly. You're going to read the book. You're going to know all of this. It's 107 content. There is no excuse for not knowing all of this content. It's a prerequisite for this course. So if you don't know this, you better get on your horse. Okay, ribose and deoxyribose, these are what? The sugars that are in our nucleic acids, right? Ribose contains an oxygen, or a hydroxyl group, at both the 2' carbon and the 3' carbon. All nucleotides have a hydroxyl at the 3' carbon. That's the 3' OH I was constantly yammering about in 107. The 5' carbon is the one that the phosphate group is attached to, right? 5' phosphates, 3' OH. These don't change between these two sugar molecules. Just the 2'. Deoxy, remove the oxygen. So when you're looking at pictures of nucleotides to determine if they're DNA or RNA, make sure you're looking at the 2' carbon and not the 3'. So don't just say this and go, oh, look, an oxygen has to be ribose. You have to look at the 2'. Okay, so ribose has 2 hydroxyls, deoxyribose only has 1. And then, of course, we need to know the purines and the pyrimidines, and you need to know which class is which, right? Adenine and guanine are both purines. They have the 2 rings. Cytosine, thymine, and uracil are all pyrimidines. They only have the 1 ring. I won't ask you to draw the structures as far as carbons and hydrogens and nitrogens and oxygens or any of that kind of stuff, but you do need to be able to recognize them as pyrimidine or purine, just like you did for 107. Oh my god! That is a giant phosphate. All right, so if we're looking at, excuse me, the 4 nucleotides of DNA, and again, how do we know they're DNA? Right? 2' is missing the hydroxyl. And if these were in a category, we'd automatically see that this is thymine, right? Deoxythymidine, which means this is the T, right? And there are no T's in RNA, so this would have to be DNA. So here's our C, here's our G, and here's our A. And as we know, this is just the monophosphate, right? Which means it has 1 phosphate group. We do know that they exist each with 2 more phosphate groups attached as a trinucleotide. So if we were talking about adenine and it had its 2 extra phosphate groups floating around, right? This is ATP, people. D-ATP in this case. And the D just stands for deoxy. Okay, make sure you can recognize these structures. And then, of course, our beloved double-stranded DNA. I'm going to point out the things that I just cannot let you guys get wrong, right? We're first, right here, no oxygen in that 2'. The 3' is the one that, right, connects. What is that bond called? Phosphodiester bond, right? Between the 5' phosphate and the 3' OH, right? 5' to 3' direction, right? 5' phosphate hanging off one end, 3' OH hanging off the other. On the other strand of this double strand must be anti-parallel, right? In this case, the 5' down here and the 3' OH here, right? We call those two strands together anti-parallel. That's what sets up this hydrogen bonding between the bases, right? We have to remember we've got phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, and the bases are what are attracted to each other by hydrogen bonding. And that's DNA, always, always, always double-stranded, except very transiently during mechanisms. Okay. If we look at RNA, it's always single-stranded, but again, 5' to 3', right? Phosphate, sugar base, phosphate, sugar base, phosphate, sugar base. And here we can see, right, here's our telltale extra hydroxyl group, which tells us it's RNA. If we look at DNA in the space-filling model, we can see that the backbone is what we can see that's spinning around and around. Okay, the double strand in here where the base pairing is, and the way it's twisted, there's a major groove and a minor groove. Okay, this is just a little picture that's showing us this. Again, 5' to 3' on one end, right? 5' to 3' anti-parallel, base pairing down the middle. I don't care about all these distances. That is, you can always just look that up. So don't memorize distances. Make sure you know about anti. Now we're going to talk about chromatin structure, right? Again, we always start with this double-stranded DNA. This is then wound around nucleosomes, which are histones, right? So please go ahead and take a look back at 107 to remember your octet of histone proteins, right? There's another histone, H1, that sits sort of outside. The DNA is wrapped around twice, each one of these histone complexes, the linker DNA in between, right? This is known as the beads on a string. So this is open chromatin. If we're wrapping down into a chromosome, and when do we ever do that? Only during prophase, right? Otherwise, we're the spaghetti shape. The spaghetti shape is the beads on the string. Okay, never just naked DNA. It's always associated with histone proteins. This coils on itself, which coils on itself, which then coils on itself. Sorry. And then coil, 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 and that bigger coil, blah, 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 until you can compress down all the way into a chromosome. Okay, and again, I don't care about nanomolar and all that kind of crap. Okay, but make sure you understand how this works. Okay, the last question, I think, that we're talking about is when they were thinking of, well, if DNA is all wrapped up in all these proteins, even open DNA is beads on a string, how does transcription happen? How do any proteins get in there to do that? And so they did some experiments where they actually use enzymes that break up nucleic acids, so they're called nucleases, and the only places these nucleases could break were between the histone proteins, right? Showing that the DNA really is attached to histone protein. And then the experiment they did, well, the DNA must be altered during transcription, any place that it's active, so that proteins to get in on the promoter, right? RNA polymerase needs to get in there, TATA binding proteins need to get in there. All those proteins that need to drive transcription need access to the DNA. If they're wrapped up in histone proteins and packed together, how is that going to happen? And so they went ahead and looked at what areas of DNA are sensitive to this enzyme, this nuclease called DNase 1, right? If they're totally wrapped up tight, DNase 1 can't get in there and cut those pieces. If they're open up in beads on a string, it probably can. And so they looked at two different types of genes, embryonic globin gene, so only expressed in embryo, and then adult globin genes, only expressed in adult. Okay, and these are the globins like hemoglobin, right? So that'd be in red blood cells. So they're looking at, right, erythroblasts are red blood cells, or they're preliminary little guys in there. And so if we're looking at treating this DNA, if we're looking at 5-day-old erythroblasts, we would see that the embryonic DNA is active, right? So we can cleave it because it's open. These guys must be closed. DNase does not cut it up. Once you're 14 days, the embryonic is shut down. Now you're considered an adult if you're a mouse. Or I guess this is a chicken, but whatever. Now the embryonic region is closed down. The DNase can't break it apart. The adult globin genes are open, beads on a spring. They can be cleaved up. And then if you're looking at a brain, right, brain cells should never be making hemoglobin, so they're always closed down. Okay, and so that set up the open chromatin, beads on a string, is where genes can be active. Closed chromatin, where it's shut down, are not active, suggesting that chromatin structure must change during transcription. And we will talk more about this later in the semester when we get to gene regulation. And then the last thing, this better be a review, the central dogma, DNA to RNA to protein. That's the direction of hereditary information. And then if we're talking about some other specialty information pathways, like some viruses, where we can go backwards from RNA to DNA and RNA to RNA to get to protein, why does this not defy the central dogma? And that's something you guys need to think about, and maybe we'll talk about sometime. Otherwise, thanks for listening, and I hope all is well. Bye-bye.